Hi, everyone, and welcome to episode 659 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am excited to be resharing our conversation with our guest, Dr. Mike Clegg, originally published on March 13th of 2015. Listeners, Mike is a renowned researcher in plant genetics and plant evolution. He has conducted fascinating work ranging from the origins of flower colors to early agriculture and the domestication of barley as a crop plant. And in our interview, Mike shared his non-traditional path to a career in science, some of the exciting research projects he's worked on, and also his life outside the lab. So listeners, get ready to learn more about another one of our brilliant people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am delighted to be here today with our excellent guest, Dr. Mike Clegg. So welcome to our show, Mike. How are you?
I'm fine, thank you.
Wonderful. Well, great to hear. And I'm excited to introduce you to our listeners today. So listeners, Mike is a professor emeritus of biological sciences, ecology, and evolutionary biology at the University of California, Irvine. He's also the past foreign secretary of the U.S. National Academy of Sciences, just recently finishing up his third consecutive term. And Mike received his PhD in genetics from the University of California, Davis. He served on the faculty at Brown University, then the University of Georgia, and then the University of California, Riverside, before accepting his final position there at UC Irvine. And Mike has a long list of career and research accolades, and I'm just going to name a few of them here for you all today. He was elected as a member of the U.S. National Academy of Sciences, a fellow of the American Academy of Arts and Sciences, a member of the American Philosophical Society, a fellow of the Global Academy of Sciences, an honorary member of the Palestinian Academy of Sciences, a foreign fellow of the Nigerian Academy of Sciences, and also a corresponding member of multiple other international academies. Now, he's also senior fellow of the California Council on Science and Technology and has been awarded the Darwin Prize from Edinburgh University. So a long list of accomplishments and accolades there for Mike. And Mike is here with us today to share his own journey through life and science. So Mike, we want to get to know you today, both as a scientist, but also as a person. So can you start off with just telling us what you like to do when you're not doing science?
Right. Well, I have multiple interests. Among them, I enjoy reading a lot, and especially history. So much of my spare time is spent with my nose in a book. But I also have had a long fascination with airplanes going back to my youth when I used to work for a crop duster in Northern California. And so I actually own and fly two different airplanes. And I love to travel and have done a great deal of it. And finally, I enjoy very much spending time with my family. I have 6 grandchildren, 2 of whom are now in college. And so I've had a wonderful family behind me.
Wonderful. Well, Mike, great to hear about how you spend your time when you're not thinking about science. And it's very interesting to hear that you're a pilot, you like to read, travel, spend time with your family, all wonderful things outside the world of science. So can you tell us a little bit about, you mentioned you like to read history. Is there a particular book, might be a history book or a different book, that you've read and really enjoyed that you'd like our listeners to check out?
Well, there's a long list of histories that I've read over the last few years, but one that I just read, which I found fascinating, is a book called Year of Meteors, which is a book about the year during which the 1860 presidential election took place, which painted a lot of the context in a much richer way than I had read before, and gave a much richer and clearer picture of Stephen Douglas as sort of protagonist of Abraham Lincoln during the election and the drive towards secession in the South. So it was a very fascinating book for me because it actually filled in some details in that part of American history that I didn't have as good an understanding of. My own family was caught up in the Civil War. 3 of my great-grandfather's brothers died in the Civil War.
Really? That's very interesting and great that you know a little bit of your family history and how it ties in with the history of our country. I like it. Now, Mike, now that we've heard a little bit more about how you spend your time when you're not thinking about science, I'd like to hear about the science next. So can you tell us how you would just describe it to someone who might not be familiar with science?
Sure. I try and describe the science I do as studying the way genes change through time and using that information to also understand the historical relationships among organisms. So that's the way I describe it. I hope that that kind of a description is accessible to most people.
Absolutely. I think that sounds very interesting. And I'd love to hear more about your research as we go through the interview. But first, I want to start off with finding out what motivates you. I know a lot of scientists have these particular, maybe motivational quotes or sayings that they keep in their mind as they go through their day to keep them focused and going. Is there one that's your favorite?
Well, I have a sign in my office that says, "This is the life." And that was actually made for me by a research colleague I had at Brown more than 40 years ago. But it's a saying that I picked up when I served in the Army 50 years ago. And it is the life. I've had a great life. It's been a wonderful experience. So that's the kind of motivational quote that comes to my mind. This is the life.
Well, Mike, I love that. I think that is a great motivational quote and a great thing to keep in your mind to make you appreciate things. You know, you might have rough days in science. You might have bad days where you're going into the office, but every morning when you open that door, you see that sign and you think, I've got it pretty good, right?
Right.
Well, fantastic. Well, Mike, next, I'd like to hear a little bit more about some of these people along your journey and throughout your career who really meant a lot to you. Maybe some of these people who were role models to you.
Yeah, so there were a lot of people who had an influence on me, as I think is true for everybody. We very much work and live in a community in science where we are stimulated by those around us. I think the best ideas I've had are the result of stimulation from other people and working together as part of a community. But one of the people who certainly had a profound influence on me was my major professor, who was himself a distinguished scientist. I was very fortunate to be able to work with him. His name was Bob Allard, and he was a member of the National Academy of Sciences. I think the third person from UC Davis to be elected to the Academy in the history of Davis. And he was a very careful scientist with high personal standards, a very skillful writer, and he had a big influence on the way I viewed science and on my subsequent career.
Well, it's wonderful to hear about one of these people in your life who really had an impact on your science and on your career and on you personally. I think it's so important as a scientist to think about these people, you know, these mentors you've had, this network that you've developed of people, because I think you're absolutely right. I mean, science is something where you realize when you look back how many people you've come in contact with, how many people have had an impact on you. And I think it's just phenomenal to be able to work in such a collaborative type of environment. So can you take us back to the very beginning now, Mike, and tell us how you got interested in science in the first place? You mentioned you spent some time in the military. You spent some time as a crop duster. How did you go towards science?
Well, I had kind of an uneven progress towards science. My father was a scientist. He was a professor at the University of California at Davis. And I was not a very good student, but I grew up in a household that valued education and intellectual activity. And that, in the end, I think, helped determine my path in life. But as I mentioned, I was a terrible student. I was actually expelled from high school.
Really? Wow.
Got married at 18. And then after 3 years serving as a paratrooper, I got out of the Army, went to the local junior college, Sacramento City College, and tried to redeem myself. The military had a big influence on me. I was successful for the first time in my life in that environment, mostly because the job I had required some computational skills that I seemed to have and many others didn't. So it gave me a measure of confidence and success that I didn't have before. And I went on to the junior college for a year or so, but then my first child arrived and I had to go to work. I worked for the sugar industry, the beet sugar industry in Northern California, and was fortunate that I got a job as a low-level technician in their research department. the process for making purified sucrose is a very interesting exercise in chemical engineering. And so I worked in that environment for about 3.5 years and learned a great deal about practical science and practical chemistry. And at the same time, I went to college part-time at night, and then later the Spreckels Sugar Company allowed me to work at night, and I went part-time in the day. And so I progressed rather slowly through the junior college. But at the end of my time there, the U.S. had become heavily engaged in the Vietnam War, and I was a beneficiary of the GI Bill, which was passed in 1967. And because of that, I was able to go back to college full-time and work part-time. So I entered UC Davis as a junior on the GI Bill and worked part-time as a research technician in their poultry department and finished a bachelor's degree in 2 years and then went on to a PhD. I didn't do a postdoc because in those days it was somewhat unusual to do a postdoc in my field. So I moved from being a graduate student immediately to being a faculty member at Brown University. So that's kind of the short story. When I entered Davis, I decided to major in the joint master's bachelor's program in plant science and agricultural economics with the goal of working in international science. In those days, in the 1960s, the U.S. Agency for International Development was fairly new on the scene. The idealism of advancing economic development in the world and addressing issues of poverty in the developing world were highly prized in the post-World War II era. The United States was hugely advantaged because it was the only major economy left standing after the war, and it could do a great deal in the world and was doing a great deal in the world, and I was attracted to that idealism. And so I progressed in that major, took a lot of economics, quite a bit of mathematics, and then I had to take an introductory genetics class as part of the plant science component of the major, and the genetics course used a set of notes on population genetics, which had been written by a different faculty member, but not the instructor, but another faculty member in the department. And they were beautifully written, and there was a fascinating example of the use of mathematics in biology, which I hadn't encountered before, and I liked mathematics and I liked biology, so I became intrigued with this. Eventually made contact with the professor who had written them, which was Bob Allard, who became my major professor. And I stayed at Davis for the PhD. It was, I think, one of the freest and happiest periods of my life. The work was fascinating. I was working on a project that was, at that time, new and fresh. There were new technologies which were coming into genetics at that time, which provided a view of genetic variation that had been unavailable to previous researchers. So it was just the beginnings of the use of biochemical and later molecular technologies and looking at diversity in populations. So for me, it was an absolutely wonderful and completely engrossing time. And I still look back on it as one of the happiest periods of my life.
Well, Mike, you have such an inspiring story. I think this is a really powerful message for our listeners out there who might be interested in science, be interested in a career in science, but think they can't do it. I mean, you went from being expelled from high school to being a prominent scientist, a U.S. National Academy member. I mean, this is phenomenal. So was there something that really kept you going? I mean, you've had so many different careers. Was there sort of a guiding theme, a guiding principle that goes through your life that sort of connects everything?
Well, probably a couple. Well, one is my situation is not quite as unusual as it seems.
I'm learning this more and more. I like it.
I know several other members of the Academy whose story is not that different from mine. So it proves that second chances sometimes work, and that one of the great things about growing up in California, as I did, is that there was a wonderful and open system of higher education that allowed you to move from one level to another and recover lost ground from the past. So I was very fortunate. I think that degree of freedom is not there today to the extent that it was when I was growing up. The commitment of the state to higher education is not the same as it was in those exciting post-war years. But my situation is not unique by any means.
Certainly. Well, very, very interesting story. Great to hear a little bit more about your own history. And I'm curious now, you'd mentioned a little bit how you got to where you are, but can you tell us more detail about one of these exciting projects that you're working on currently or have been working on recently?
Yeah, I'd be happy to. So in my career, I've sort of moved through maybe 4 or 5 different phases and asking rather different research questions, some of which have been more successful than others. But the first major phase for me had to do with a controversy which was raging in the field of population genetics in the late 1960s and early 1970s. And population genetics began as essentially a mathematical consistency argument, which was developed during the period of the late 1915 to 1930 or so, to show that the particulate inheritance, which had been discovered by Mendel, was consistent with the theory of evolution by natural selection posited by Darwin. And this mathematical argument began with the view that you could treat individual genes as kind of the atoms of heredity, and you could abstract down to the level of a single gene and ask how that gene might change in frequency over time, and use that as a way to provide a theoretical explanation for the evolutionary process. Well, one of the problems with that abstraction is that genes, in fact, don't come as particles.
Right.
They're linked together on chromosomes. And so different genes may be inherited together because of their physical linkage on chromosomes. But over time, they become randomized with respect to one another through the action of genetic crossing over or recombination. So the issue which was important in the late 60s and early 70s was to ask whether the way in which genes interacted and selection on phenotypes, which are the result of interacting genes, might actually act to retard this process of randomization at the statistical level. And I did some early work shortly after I finished my PhD thesis on plant population genetics, which showed that in self-fertilizing populations of plants, barley in this case, that you tended to see the evolution of correlated complexes of genes. That is, the effective recombination was retarded over time. And so this had a big impact and helped sort of launch my career.
Sure.
And after that, I wanted to follow up on that. And in order to follow up on it, I switched my research emphasis from plants to the fruit fly. It was possible to compose Drosophila populations and watch how recombination operated at the population level over time. And there's a simple null hypothesis from population genetic theory that said that the associations among genes ought to decay at a rate which was proportional to the recombination rate. So it was possible to ask whether that actually was the case in populations. And it turned out not to be the case in these experimental populations, but in fact that the rate of decay was accelerated. So they went towards randomization much faster than predicted. And I followed that up with some theory, particularly with computer simulation work where it was possible to construct computer models of chromosomes that, for one imagined you had now 100 genes on a chromosome, and you could actually mimic the whole genetic transmission and recombination process in the computer simulations and ask how that behaved if you used good estimates of the selection operating on whole chromosomes. And it turned out that those results exactly corresponded with the experimental results from Drosophila. So that had a significant impact and is work that I am still proud of. It led to a series of papers in the 1970s and into the early 1980s. But at the end of the day, I'd sort of answered the question that I had set out to deal with. And
right. So, where to next, right?
Exactly. So the problem, what to do next, emerged. And I had a colleague named Jim Rawson, who was a molecular biologist. And at that time, this was the sort of mid-late 1970s, every day it seemed like there was a wonderful discovery in molecular biology. I can remember having lunch with Jim one day and him telling me about the discovery of intervening sequences in genes, something that hadn't been known. And
wow.
This whole powerful molecular technology suddenly gave us a fine-scale view of the nature of genes that we had not been able to dissect before based on classical genetic approaches. So in a way, our whole knowledge of genes at the microscopic level was wrong. And even though macroscopically Mendelian genetics worked fine. So Jim had a big influence on me. Jim and I began to collaborate on applying the tools of molecular biology to understanding gene evolution. And this was a very hard job. This is the late 70s, beginning of the 80s, because the technology was very crude. And at that time, nobody had cloned a plant nuclear gene. And we set out to try and do that and did not succeed. But Jim at the time was working on the physical structure of the chloroplast genome in Euglena, which is a protist, but it has a chloroplast. And the chloroplast is the cellular site of photosynthesis. It's an organelle which has its own DNA molecule. And the chloroplast is the result of an endosymbiotic association between a prokaryote, a cyanobacterium, and the early eukaryotic cells. And all photosynthetic organisms derive from that kind of association, including all green plants. So the chloroplast genome is quite abundant in plant cells. It was much easier to clone chloroplast DNA in those days and to work with it and to purify it. And so we began to study the evolution of the chloroplast genome and to apply that to understanding plant relationships. And that turned out to be a very fruitful line of inquiry that I worked on for about 20 years. And some of the big results were that we discovered that the chloroplast genes evolved a good deal slower than nuclear genes in plants for complicated reasons. But that opened up the possibility of using chloroplast gene change as a way of understanding organismic relationships in plants at the very deepest levels of evolution, because the rate of mutation was slow enough, the rate of mutational accumulation was slow enough that you could look at deep time levels. And so we later, with another colleague, I spent a wonderful sabbatical year in Australia at CSIRO, about 81 to 82, and began to work with a man named Gerard Zarowski, who was a very early person working on sequencing chloroplast genes. And Gerard taught me sequencing technologies, and we continued to collaborate for a number of years. And we were able to begin this process of using a particular gene on the chloroplast genome as a tool to understand plant organismic relationships. And this gene is actually responsible for fixing CO2 in photosynthesis. So it sits at the very nexus of the Earth's carbon metabolism. Very interesting gene. And so we began sequencing that gene. It got picked up by many others and used as a tool to understand plant phylogenetic relationships. So phase 3 of my career was focused on studying the evolution of genes that determine flower color in plants. And in particular, the plants that we chose to study in this regard are the morning glory, which is a very diverse genus. It has about 600 or so species. The one species called the common morning glory got a wonderful diversity of flower color differences, I think largely because of early human selection. These plants are vine. It seems very plausible after the domestication of maize around 10,000 years ago. Of course, like all farmers, the early Neolithic farmers would have been plagued by weeds, and one of those weeds would have been morning glories, which as a vine grew up the maize stalks. And they probably selected and saved just for aesthetic purposes different flower color mutants. And so this wonderful diversity of flower color mutants in the morning glory probably trace back to selections by early Neolithic farmers here in central Mexico.
Very interesting.
So the hope was, of course, that we'd be able to dissect this process and understand exactly which mutational changes were responsible for the changes in flower color, and possibly link those to the way in which insect pollinators visited different colors. And we'd shown in other work that the insect pollinators do have a preference for different colors. And so the idea was that we could link the whole process of natural selection from the behavior of the insects all the way down to the genes which were being influenced by this insect selection of different flowers to visit. Well, it turned out that that vision has still not been achieved. We learned a lot about the evolutionary genetics of the flower color polypathways, and we learned, for example, that many of the genes come as multi-gene families, that the genomic processes of diversification are much more complicated than this sort of simple-minded hypothesis. And so it's still in many ways an incomplete piece of work, but we spent about 20 years working in that area. And then the final phase has been devoted to using genetic tools to try and understand the history of plant domestication. And this has been done mostly with barley and also with avocado here in Central America. So plant domestication is the sort of one of the key events in human evolution, began in earnest 10 to 12,000 years ago. But one of the kind of remarkable things about plant domestication is that it took place apparently independently. That is, humans learned to domesticate plants independently in several different parts of the world, more or less contemporaneously, around 10,000 years ago. So you had wheat and barley in the Middle East. In the Fertile Crescent region, you had rice in Asia, you had maize in Mexico, and other actually plants also being domesticated in different parts of the world independently, but at about the same time by humans. This was, of course, responsible for the transition to complex civilizations and more complex economies. And so one interesting question is, why in the world did this happen at the same time in different parts of the world and evidently independently? And nobody really knows.
Right, one of these great mysteries.
Yeah, it's one of the fascinating mysteries of human evolution, but probably a plausible speculation is that it's linked with climate change, that this is the end of the Pleistocene glaciation. Climates were much more variable from year to year, and therefore food supplies were more uncertain. And this may have driven early human hunter-gatherers to save and plant seed as a kind of insurance against the fluctuations in the climatic environments that they were living in. But another question, and the one that we've been more interested in, is if you take a plant like barley, which was one of the first cereal grains to be domesticated by humans, did this just happen once? Certainly a parsimonious view of a complicated process like plant domestication would suggest that it might be unlikely to happen independently more than once. And so it's been possible now using genomic tools to ask these kinds of questions, because you can ask whether different primitive domesticated forms of barley had different origins from the geographic diversity of wild barleys. And the answer to that question appears to be that it did happen more than once, that it happened at least once very clearly in the Fertile Crescent, but likely more than once, perhaps a second time, east of the Zagros Mountains on the plains that now constitute present-day Iran, or in the intermontane valleys of the eastern part of the Zagros Mountains, so that even though this seems to be an unlikely event to happen repeatedly, the evidence suggests more that for whatever reason humans were driven to domesticate a major grain crop like barley independently in different geographic locations.
Mike, this is fascinating research and great to hear about these different projects you've been working on. And I feel like I could listen to you for hours. I love how you tie in the history behind the science and how it all fits in with your research. This is just incredible. So great to hear about some of this amazing work you've done in science. And I think we've hit a lot of the high points in your research, in your career. And we haven't yet touched on too much some of these struggles that I think scientists face. And I think every scientist, regardless of how great the job is, how wonderful it is to be in science, you have days where it seems like things just aren't going your way. So can you tell us a little bit about one of these times where something just wasn't going your way? You were struggling with something, or maybe you had a major failure, and how you fixed it?
Well, I think one of the keys to success in science is persistence. There are a couple of things that are important. One is a good taste in questions. You've got to ask questions that really have empirical content that you can expect to get an answer to with the tools at hand. I think in my life, there's been a constant evolution of technology, which has provided more and more powerful resolution of the sorts of problems that have interested me. And so over the years, it's been possible to address some questions today that did not have real empirical content when I began in my career. So one key is to try and be sure that you're asking questions which have empirical content in the context of the tools that are available to you. But the other is persistence. Things don't always work, and you've got to keep working on trying to find ways to get the technology to give you the answers that are hidden away. Many of these methods have evolved enormously. And when I first began in the DNA sequencing business back in the early 1980s, the primary method was a method which was called Maxim Gilbert sequencing. It was very cumbersome. It took about 6 months or so to sequence a gene because of the complicated strategies involved. Nowadays, you can sequence an entire genome in a matter of a week or less.
Right. Drop it in the mail and have someone else do it too.
Exactly. And you don't even have to do the work.
Right.
So things evolved a lot, but being persistent, trying to make these methods work for you to solve problems is extremely important. I also found that it's important in other aspects of life. So it's important not to give up too easily. But, everybody has failures. And one of the ones that for me was that this vision I had of the flower color work never really came to fruition. We never really got to the point where we could construct a convincing argument that traced the causal pathways from insect pollinator behavior on different flower colors down to mutational changes in individual genes. We just didn't get there. And nevertheless, it's been an interesting problem which has revealed a lot along the way. But sometimes you just don't get to the final answers that you had hoped that you would. But if you're adaptive and creative, you can usually make good things out of what you do learn.
Absolutely. Well, Mike, I think you brought up so many important points here. I mean, I think persistence in science is so important, and making sure you are sort of picking the right questions. I think a lot of it is sort of planning and thinking it through, but I think sometimes it just comes down to luck. Sometimes you talk to different scientists, the things that they choose, it's sort of happenstance the way things worked out, and you end up in this particular field working on this particular thing, and it happens to work out, whereas some projects just don't work out, like this flower color one where you were never really able to get to the answer that you were seeking. So great points you brought up there. Now, we talked a little bit about your interest in travel, and I know you've been able to travel quite a bit. You're associated with a lot of international academies, things like this. Can you think back, Mike, on all the travel you've done for science over the years? And can you pinpoint one particular place that was just your favorite place that you've traveled, and tell us a little bit about it?
I don't know that I could pick a particular favorite place. I have many favorite places that I have enjoyed going to, but some which have been remarkable experiences. And I guess one I'd pick out in that context is a trip to Iran that I took around 2007. I spent about 10 days in Iran with a delegation of people mostly from the National Academy of Sciences. We were involved with a joint project with the Iranian Academy of Sciences. which still goes on to this day. Iran is a fascinating country, and the level of educational attainment is really pretty high. We spent a fair amount of time at a major university called Sharif University in Tehran, which is roughly the sort of Iranian equivalent of MIT. It has a very strong engineering basis and strong physics and so forth. One of the interesting things in visiting Sharif University was to discover that most of the graduate students in physics were women, unlike U.S. physics programs.
That is very different, yeah.
In fact, in Iran, the gender proportions are fairly heavily biased towards women, so more than 60%, if I remember correctly, of college students in Iran are women, and that's a source of some concern to the Iranians. But it's an interesting commentary on what we think of when we think of a country like Iran and the realities that you see on the ground and the way in which social evolution takes place in different countries. So the experience in Iran was interesting from that point of view. It was also interesting from the point of view of my work on barley domestication, because Iran might have been one of the other areas of domestication of barley. And finally, we spent a wonderful time visiting Persepolis, where you could see this wonderful history of culture going back more than 2,500 years, which had a great efflorescence and influenced some of our own cultural history. So Iran was quite interesting. And it allows me to make a different point about the work with the National Academy of Sciences. Over the years that I was Foreign Secretary and continuing on beyond that, one of the goals has been to use science as a bridge between nations in conflict. And so despite the fact that the U.S. government and the Iranian government were often at odds, we were able to carry on a very healthy and stimulating scientific exchange with the Iranian scientific community that actually was encouraged by the governments on both sides, because it provided a kind of low-level bridge that allowed for cooperation and communication despite higher-level animosities. And science can often work in that way. So we try to use this model of science as a bridge in building relationships more generally in our global work. And science academies turn out to be a nice way to do that, because most countries have them. And so you can sort of think of every country as having a kind of node, which is a science academy. Often the science academies in different countries include the leadership of science in that country. And so there are ways of stitching this all together into a global network to work together on common global issues like climate change or emergent diseases or water resource problems or any of advancing the status of women in science, concerns about the quality of science education. All of these things can be addressed on a global basis. And the science community is a great vehicle to do that, because we all share a common culture in science, and we're able to communicate based on this shared sense of interest in scientific problems and a shared sense in our participation in the history of science. And so that brings us together as a single community that is able to, I think, affect positive change in the world.
Certainly. I love this analogy you've drawn of science as a bridge between countries, between cultures, and really being able to, as a result, take a global approach to some of these big problems that the world as a whole is facing today. So wonderful description there. And I really like how you're describing just some of the social and cultural differences that you see when you travel. And I know this is one of the reasons I love going to different places, is you get to see how people live differently across the world and think about how it compares to, it just sort of makes you think about things at home that you normally wouldn't think about, right?
Exactly. It also makes you appreciate that we're all the same.
Absolutely. Well, Mike, I love hearing about these travel experiences that you've had. And I think as a scientist, you get a lot of great opportunities to travel and a lot of great opportunities to work with incredible people over the years. And I know scientists have some stereotypes out there when you see scientists portrayed in the media, perhaps on TV and movies, things like that. And I like to break those stereotypes on our program. We always like to talk about how scientists are perhaps not what you would expect. There are a lot of serious people who are focused on research, but they're also a lot of fun. You have these fun traditions, these quirky, funny memories that spring up over the years. So can you share one of these traditions or one of these memories with us today?
Well, one of the things that characterized my working environment from the beginning is always the sort of camaraderie and community spirit of it. So in the early days when I was at Brown University working on Drosophila and learning the technologies of Drosophila genetics, I had a wonderful pair of colleagues, Jim and Margaret Kidwell, and we would always have coffee together each morning and talk about scientific problems and enjoy this sort of relaxed, free exchange. At that time, Margaret was actually finishing her PhD thesis, and she was working on a very puzzling phenomenon, which was one of the peculiarities of Drosophila is that you do not observe genetic recombination in males. But she was finding patterns of recombinant chromosomes in males, along with high frequencies of lethal mutations. And ultimately, she was able to describe this as a syndrome called hybrid dysgenesis. And it turned out that the basis of the syndrome was an invasion of Drosophila melanogaster, the genome of Drosophila melanogaster, by a family of transposable elements, which got called P elements. But all of this, trying to understand this complicated phenomenon, these collections of observations in this relaxed environment of having coffee and speculating, was part of something that's continued throughout my career. So in later years, for example, at UC Riverside, when I moved there was a great tradition of going to the local pub. There was a pub on the campus called the Barn, which we would go to on Friday nights and sit around and drink beer and talk about whatever the issues of the day were or interesting scientific questions. And I usually did that with my graduate students, but also with my colleagues from UC Riverside, and many of them were working in different fields, but there was this opportunity for cross-fertilization of ideas, understanding other people's problems and questions in this relaxed and stimulating environment. And we continued that tradition at Irvine by having lunch together as a lab almost every day, and once again, talking about whatever was of interest at the moment, but in an environment that provided both stimulation and it was an opportunity to express crazy ideas without feeling that you'd be censured for them.
Right. It's not in the lab, so it's okay. Yeah.
Right. That's not quirky, but it's been a very important part of my history as I look back on it. And I can't really think of anything that I would label as exactly quirky. I'm sure there are quirky things, but they don't come to mind.
Right. Maybe you're just a quirky person, so you don't notice them, right? Well, Mike, I think this is a great example of some of the fun activities that scientists do together, is sort of getting out of the lab, spending time together, either during coffee breaks, at lunch, during some of this pub time, and just talking to each other. And I think this is something that, a lot of people might not realize, that science is such a communal and collegial sort of career. You spend a lot of time together, and I love this description of kind of throwing out any crazy idea out there without worry that people are going to laugh at you, or at least you can laugh together at it.
Right, exactly, yeah.
Well, and I think, while we're on the topic of crazy ideas, these big questions that you would love to answer, one of my favorite questions to ask scientists is kind of what the big questions are in their minds. So can you tell us, Mike, if we took away all the normal restrictions on science, if things like funding and staff and technology and even feasibility were not an issue for you today, right now, what is one question that you are dying to know the answer to still?
Well, I think I'm very much influenced by what I'm doing at the moment. And so what I'm still doing at the moment is worrying about the domestication of plants. And I would like to know in much greater detail, geographic detail, what the potential sites were of barley domestication by Neolithic peoples and how that tied in with the development of culture and civilization. One of the things that I find quite stimulating, and it's outside my own research, but it's using the same principles, is the wonderful work on human evolution that has grown out of the genomics era, where we have a so much richer view of the details of human evolutionary change, because we can use the genes to trace the past of human populations. And it's understanding how that interacted with the domestication of plants that I find a fascinating question for the future.
Great question. Well, Mike, in addition to talking about some of these big questions, I do like to ask scientists about advice. And I think as a scientist, you get a lot of advice throughout your career from a lot of different sources. Can you pinpoint for us today what you would consider the best piece of advice that you yourself have ever received?
Well, I spent 6 years as the dean of a complicated college at UC Riverside, which had a lot of different moving parts, and enjoyed it. I found it a wonderful challenge and interesting to try and think about the future of an organization. But I can remember that I got one piece of advice from a very distinguished scientist who had been a figure in my early career, and in fact, even in my childhood, who said I shouldn't do it.
Really? That's probably the last thing you wanted to hear right then, right?
And you do pay a price for that kind of thing, in the sense that it slows down your scientific productivity, probably inhibits some of your grant success and so forth. So the question was whether the price was worth the challenge. I would say that it was, but that's a piece of advice I didn't take and wonder about.
Do you have any regrets, Mike?
No, I don't think I have any regrets. I've had a wonderful career. In fact, as I look back on it, I can't imagine how I could have been so fortunate. I chose a career in science at a very good time when there was still an upward trajectory in the United States, because higher education was growing a lot still in that era. So there were plenty of opportunities. But there was also an era of incredible discovery, and to be a small part of that was a wonderful experience and very stimulating. I can't imagine a better life than the one I've had.
Well, it has been a delight to hear your story, Mike. Is there any other last piece of advice, or maybe one last piece of inspiration that you want to leave our listeners with today?
Well, I don't know about inspiration.
Maybe anti-inspiration, whatever you'd like to throw out there.
I think that it's wonderfully enriching to know more about the world that we live in. And science gives you that window. It makes the experience of life much richer and much more interesting, because it gives you a more detailed and more beautiful understanding of the physical and biological world that we're a part of. So even though I don't know a whole lot about it, I think one of the most fascinating things in the last 30 years has been the sort of revolutions in cosmology, which have given us a much deeper and in some ways more challenging view of the universe that we live in. And so science gives you this opportunity to experience life not only in the present, but in the past and across great spans of space as well. And that's something which is priceless.
Well, very well said, Mike. And can you tell our listeners, if they want to learn more about your science or about you, how can they find you?
Well, I don't have a web page or anything like that, but they're certainly welcome to contact me via email. And my email address is mclegg@uci.edu. It's very simple, my first initial, last name.
Wonderful. Well, listeners, feel free to reach out to Mike if you have any particular questions. And Mike, thank you so much for joining us today and sharing your phenomenal story.
No, it's been a pleasure.
Well, it has been a pleasure to chat with you as well. And I want to thank our listeners out there. We'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.